All right, good afternoon, everyone. So today you're going to have a lecture on stability and processing of precursor proteins. And you are very lucky to have an expert in the field, and he's actually an expert in the field for many years. Just briefly to mention what he has done. So he was the person who actually discovered seven of the nine enzymes belonging to the protein convertase family, which are important for a wide variety of biological processes. And in particular, more recently, he has discovered that a point mutation in one of these proteins, which is called PCSK9, causes dominant familial hypochondroplasia. So he's a very productive scientist. He works at the IRCM. He's a professor at the IRCM for several years, for many years now. And he has received many awards during his career, so you should be aware of that. And I just mentioned few of them. He's fellow of the Royal Society of Canada. He's officer of l'Ordre du Québec. He's member of the Order of Canada, and so on. So, well, I think you have really the expert in the field for this lecture, and I hope you will really enjoy this lecture.
All right. So I'm told that we're going to give the talk in English. So si vous avez des questions en français, il n'y a pas de problème. Mais ça va se faire en anglais. On m'a dit que quand on donne une conférence, la meilleure chose à faire, c'est vous donner directement la réponse. C'est-à-dire vous donner exactement ce que je vais faire. Ce que j'aimerais que vous appreniez, we're going to switch back to English. What I'd like is really to learn when you come out of this 2 hours that we're going to have together, I want you to learn a few things. Maybe it's a new word for you. It's a word that you've never seen before. But I hope that after 2 hours you will really know what is a secreted protein, how does it get secreted, what are the forces that change its shape before it reaches its either final destination or when it's secreted. I want you to understand that. This is the basis of the whole talk. Right? So, I think you will receive all of us, we're going to get it by email. I don't many of the slides of this talk. But I'm giving you some of the references that you may want to read. Some of them are review articles. It's very exciting because we are really now at an era where we can discover genes, and actually within about a decade, within about 10 years from the discovery, we can go from bench to bedside. We can actually go to the patient and find out whether it can help the patient. You're going to see some of the stories I'm going to tell you today. I'm going to make it like a story. I'm not trying to get you to learn everything by heart. That's not the point. The point is to understand that the protein, when it enters the secretory pathway, it never leaves in the same shape as when it enters. It undergoes modifications. And that's what we're going to be talking about. So these are the type of references that give you an idea of what happens with these modifications, and we'll deal some of these questions that I'm going to be giving. So that's the plan. When you come out of this talk, 2 hours later, some people, I used to get this some years, is this coming at the exam? The point is not to give you an exam to make you fail. The point is to examine you whether you understood the basic premise that a protein has to undergo various modifications, either post-translation modification or cleavage, before it reaches final stage. I want you to understand that. And once you understood what kind of modifications are available, you'll appreciate how our genome has been diversified over the years. So, we'll first understand what are the modifications that the protein can undergo. We'll give you some survival on this. And then we're going to concentrate on a family that I spent 25 years understanding this family. We've cloned many of the members of the family, and we're going to see how not only we're understanding their structures, but now we can begin to modify them in diseases. And it's very powerful. You can actually treat a number of diseases by modifying these enzymes. And I will try to tell you some of the diseases and how they are being treated right now. As we said, we have a clinic right here, about 100 meters from here, and we got about 300 patients being treated against some of these convertases every day. So, first of all, a definition. What is a secretory pathway? I hope you understand that. And when I get this sometimes over the years, and people start thinking, oh, it's going to the nucleus, oh, it's going to the to whatever other compartment. The important thing, when we talk about the secretory pathway, we're talking about a protein that's going to leave, that's going to be translated by an mRNA in the endoplasmic reticulum, inside the lumen. Okay, it's going to go inside. It's going to leave the endoplasmic reticulum after it underwent a number of modifications. And then it's going to go into these top two vesicles, and it's going to move through the lamellae of the Golgi. Okay, this is a complicated story. There's a number of new beautiful structures now beginning to understand how they continue this Golgi. There's a number of Nobel Prizes won because of this structure here. And once it finishes with the Golgi, it's now ready to be sent to its destination. It can either go to lysosomes, it can go to the cell surface, many receptors and various glycoproteins stay at the cell surface, or it can actually be stored in very dense secretory granules. You had your lunch today, after eating your lunch, you have to digest your lunch, you have to get rid of the excess glucose that you had, dessert or something like this that you've eaten. So the pancreas has to begin to secrete insulin to allow you to digest out the food that you have. And so insulin is going to be stored in these very important granules. They're called dense core secretory granules. And once this body or the pancreas in those cases senses there's a lot of glucose because you've eaten, so say, I'm going to secrete this glucose. It's called the regulated secretory pathway. It's going to spit it out, and that will go to the liver and say to the liver, hey, there's a lot of glucose circulating, take it out, because I want to make energy out of it. I'll make ATP out of it. So this is called the regulated secretory pathway. It's usually a relatively slow one. It takes 2 to 4 hours for the insulin to be made and to be secreted. But sometimes you need to get your message very fast down, within minutes. And the way you do it is something called the constitutive secretory pathway. You want it to go, you don't have to go through all this process of making dense cores and so on and store it. You don't want to store it. All you want to do is just send your signal out. And that's called the constitutive secretory pathway. This is where all receptors, growth factors, and secretory proteins go through. They're just continuously pumped out once you have a need. So this is what we're going to be talking about. So all the proteins in the secretory pathway are going to pass through these lamellae. They're going to make a decision here. There's a decision that has to be taken. I don't have time to detail to you how the decision is made. We just uncovered that last year. But there's a decision that decides that you go this way or this way. Once the decision is made, the protein is going to continue, get its final form, and get ready for secretion. That's what it is. Right. So I just want you, it's coming Christmas, so I want you to make some kind of comparison for you. That here's the Christmas tree. You're going to go buy your Christmas tree or whatever, or if you have a chalet or whatever, you can actually cut it out. Fact is that this is how a Christmas tree looks like. But many Christmas trees look like this. What's the difference? What makes you and I and so on a different one from each other? It's the modifications. Okay? You can now dress that Christmas tree in such a way that each one of us will have a different taste, have different bowls and whatever. So once you've decorated your tree, each tree is going to be looking different. Imagine the proteins that are going through the secretory pathway. They start like this, and when they leave, they're like that. So you can imagine that each protein will be decorated differently, and that's what we're going to be talking about. What makes a protein different from the other? What makes an elephant or a Drosophila different from humans? It's the decoration. For secretion. Now this is it. We estimate around 2,000 to 2,500 proteins that are going to go through we have the genome, which is about 20,000 proteins. No, each one of us. At least 15 to 20% of them are going to go through the secretory pathway. People are calculating even 30%, up to, let's say, 6,000 proteins are going to go through the secretory pathway. Of these, they're going to go to different destinations. Okay? So what happens when it first, and there must be certain signals that decide that this protein is going to be secretory or not. So what are those signals? That's what I want you to learn. What makes a decision whether you're going to stay in the cytosol, go to the nucleus, go to mitochondria or other destinations? These are protein signals that will tell the cell whether or not you should go this way. If you have the right protein sequence that tells it where to go, in general, this is called a signal peptide. It's a sequence at the N-terminus of the protein that is highly hydrophobic, that can cross the membrane, and then it will begin to meet other proteins inside the lumen of the endoplasmic reticulum. So these proteins are going to say, I want to make a disulfide bridge. I want to begin to show that you have a correct folding, you have dips, you have what is known as molecular chaperones that will make sure that you are correctly folded. You have proteins that will add sugars that will now, if you want to start making the decoration of your protein, one of the first decoration the protein undergoes is sugars additions. It's called glycosylation. Okay? That's the first thing. Within a few seconds, as the protein gets into the lumen, you already have the addition of this glycol phosphate, which is added on block to the protein set. Every time it meets, and I'll show you what specific motif, it will directly dress it with a sugar. So it will meet the number of proteins as it gets in. This is the first two seconds of synthesis. So the first question is going to say, do you have a single peptide? Yes, I do. If you have a single peptide, you either get in. If you do not have a single peptide or other sequence I'm going to show you, you just don't get in, you get out of it. You're not a secretory protein. So, conclusion is that secretory proteins have specific signals that define them as secretory. One of the major ones is called the signal peptide. So what is the signal peptide? This is the N-terminus of the protein. It's beginning to be translated from the mRNA. Okay? It usually has a relatively short, about 1 to 5 amino acids. Then after there is an alpha helical region. This is usually a very hydrophobic region. This is what allows it to get in. Okay? This hydrophobic region is between 7 and 15 amino acids. We've seen signal peptides up to 30 amino acids. But the majority of signal peptides are about 7 to 15 amino acids of this helical structure. Once this helical structure has been recognized by a system in the cell that says, you're a secretory protein, you get in. Now you've done your job. You basically got to the address. The address was, get me into the ER. You got it. Once you've done your job, we can get rid of you. We don't need you anymore. You've done your job. So there is a system that will start cutting out that signal peptide. Very efficient. See? And the signal to cut it is usually within a region of 3 to 7 amino acids. And it has to be a very small amino acid. Small amino acid, X small amino acid. Usually an alanine, serine, glycine. This is the cleavage site of the signal peptide. So all proteins that are secreted, you always find helical region, small stretch, and then the small sequence, which is the recognition sequence of the signal peptidase. It's a complex of proteins that will cut out the signal peptide. Now, I want you to learn a few nomenclatures here because people sometimes don't understand what this means. Usually a protein with a signal peptide like that, for example, it can be membrane bound, which means it can have a signal peptide, but then later on it will have a transmembrane domain and then a cytosolic tail. Or it can be soluble, it has none of them. Just a signal peptide, it goes into the lumen of the ER and gets secreted. But if it's membrane bound, there are three types of membrane bound secretory proteins. To know that. Okay? First one is known as a type 1 membrane bound protein. It means it has a membrane domain close to the C-terminus, to the end of the bond. That's called a type 1. Another protein could have the same transmembrane domain, except instead of being at the C-terminus, it's at the N-terminus. So here's the N-terminus. Often it doesn't have a single peptide, this type of protein. It's called a type 2. So it gets in, here's the helical region. That's the signal to say, get into the ER. It goes in, and it has a luminal part, which is now inside the endoplasmic reticulum, which will be exposed outside of the cell. So there's a number of proteins called type 1 and type 2. You will see some as we go along. Another one, which is another added stuff during development, during evolution, is what is known as a GPI anchor protein. It's a protein that is usually soluble, but it has a specific sequence at the C-terminus that is added to it as it traverses the ER and to the Golgi. And it's called the GPI anchor, glycosyl phosphatidyl inositol anchor. That makes it highly hydrophobic, and it's bound to them. So you can see that this is the simplified view. The simplified view of a single transmembrane path is either a type 1, type 2, or the GPI. Many proteins, some of you may be working on receptors and GPI receptors or GPCR receptor as it's called. They may have 7 transmembrane. They may have 5 transmembrane domains. You can have a number of transmembrane domains. That's another story. Okay? The point is you must have at least one to get into the ER. Otherwise you won't get in. You either have a signal peptide or a transmembrane or both. Right. So what are the decorations that the protein undergoes? Okay. So if you calculate you and I in this audience, we have about 20,000 genes, give and take a few. How do we get from 20,000 genes, when it grows off enough, and just a little fly has 16,000 genes, it's a little fly. And we are 20,000, not too much more. We are human, we are much more complex beings. How do you get to this complexity? How do you diversify to give you this high complexity? Well, one way of doing it is by what is known as alternative splicing, because from the gene you're going to get an mRNA. So you can have this mRNA spliced differently, so you can actually produce different kinds of protein out of the same gene. That's called differential splicing. You may have heard it in your course. Or you can get alternative promoters. You can say a protein can start with a different promoter. The gene will be translated, and you can get different proteins. Or you can get what is now known as mRNA editing. This is exactly what the HIV virus uses, exactly to fool the cell by editing its HIV genome. So this kind of diversity goes from 20,000 genes to about 100,000 different proteins that can be produced out of 20,000 genes. So you multiply your diversity by 5. Right? But now, where the big diversity occurs is in the post-transition. I'm just showing you a few. There are 200 post-transition replication. Imagine, you have in your Christmas tree, you have a few balls and shiny stuff, but you have 200 different ones. Imagine how your tree would look like if you use the 200. I'm going to concentrate today, and this post-transition modification goes now from 100,000 to more than 1 million. We've now added a factor of 10. So 10 times 5, 50-fold more genes we started with than what we started with. That's what makes the diversity in our genome. We're going to concentrate on 5, because it's just too much. I finished the book, I just wrote a book on this. You know, it's very complex. Each one of them has its own effect. But most of them are reversible. That's the important point to remember. You can add your ball on your tree, but you can remove it, right? But if you cut a branch of your tree, you cannot put it back. It's finished. It's cut. Right? So it's the same thing for proteins. You can do sulfation, you can do lipidation, you can do N-acetylation, but the only one that is really irreversible is called peptide bond cleavage. If you cleave a protein, you cannot put it back ever again, as we say. You know? Now, so remember that peptide bond cleavage is irreversible. If the cell decided to cut a protein, it will be cut essentially into 2 pieces. All right, so let's start with the first decoration. The first decoration is the sugar. Within a few seconds as the mRNA is being translated and they elongated this structure, the machinery inside the endoplasmic reticulum will scan. Every time it sees a sequence asparagine X, could be anything except cysteine here, asparagine X, serine or threonine, every time it sees that, it says, oops, that's the site of N-glycosylation. N, why? Because of asparagine. This is the abbreviation for asparagine. N. So the N-glycosylation is always NXS or T. So you can check your protein, check your number of NX or T, and you realize that your protein is glycosylated. I want to give you a small story. A woman who, I don't want to say her name, she got a Nobel Prize, right? And she just published a wrong paper. She thought she had a protein that goes to the nucleus. She didn't look for NXT. She was wrong. There were 10 NXT in her protein. Her protein was not alone going to the nucleus. Her protein was a secondary protein. We just got the paper accepted. I just want to tell you, be careful when you look at the protein. Check for the NXT. This is very important. Okay? If you have one, or a secret, as long as you have the secret peptide, it will be N-glycosylation. Now, what is N-glycosylation? N-glycosylation adds en bloc a whole series of sugars. It's always the same series of sugars. It's 2 N-acetylglucosamine, 9 mannose, and 3 glucose in series. So this addition in block decides that there is an N-glycosylation site. There can be many of them within the same protein. What does this N-glycosylation do? Well, it changes the structure because the size of each N-glycosylation is about 1,500 Daltons. It's quite a bit. You know? It's like the equivalent of about 10 to 12 amino acids that you're just putting on one amino acid. Imagine the size. Okay? So you can now decorate your protein at different N-glycosylation sites. And there is a drug that you can isolate from mushrooms. It's called tunicamycin, which blocks the enzyme. It blocks the enzyme. What happens when it's N-glycosylated? Well, here we go. The protein is coming into the cell. It's being translated. There is an NXXOG. Poof, it adds the structure that I told you. 2-N-acetylglucosamine, the mannoses, and the glucose, cereal. Or block, added to the protein, right there. But that would remain like that. Now it begins to remodel. There is a whole remodeling program, and a number of enzymes are involved. Here at McGill University, there's a number of ladies, there's 2 ladies who discovered these enzymes, who actually cloned them. And we know that we can start with this on-block one and end up at the end with a much different protein than what we started with. There's a number of glycosidases, fucosidases, a number of sugar-modifying molecules that will be added to give you the final form of your sugar. Now you may say, well, why is that important? Well, now is the season when everybody gets flu, right? You get a flu vaccine and you get. And what do you think the virus is using? It's exactly that. The virus is using that to get into your cell. And when you buy the Tamiflu that you've heard about a few years ago, that all of us got vaccinated and so on, here in Quebec because of the H1N1, H1N5, it's because we're blocking the addition of sialic acid and these sugars, and we're preventing the virus from coming in. The virus knows that, and it exploits the ability of the cells adding these sugars to get in and infect you. So don't underestimate the importance of these sugar additions. Now, that's one major glycosylation, and glycosylation is a major modification of proteins. In fact, we estimate around 80% at least of the proteins that are secreted are N-glycosylated, 80 to 90%. Another one, which was underestimated until a few years ago, when the group in Denmark cloned all the enzymes that make O-glycosylation. These are now, instead of being glycosylated at asparagine, they're going to be glycosylated at serine or threonine. It's called O because of the OH of the serine. These are much smaller sugars. There's about 5 pieces as opposed to 20 pieces in the N-glycosylation. And it modulates the function of secretory proteins. Okay? It affects the stability, the solubility of proteins. If you look at your saliva is full of O-glycosylated protein. That's what makes it so smooth. That's what makes it that you can digest food and it runs. It would be very tough if you didn't have the O-glycosylation. In fact, we can create mice that don't make these enzymes, and their saliva looks like a piece of wood. You know, you cannot get smooth saliva if you don't have O-glycosylation. Just an example. This is the kind of protein that get O-glycosylated, for example. The protein I was interested in is propiomyelanocortin, the precursor of beta-endorphin. You get FGF23, your bones will not be developed if they're not O-glycosylated. It's a big game here, and I'm not going to talk too much about it, because there's a speaker coming on Monday who will discuss this story. Is that this O-glycosylation site actually prevents cleavage of this precursor. It stabilizes the cleavage site, so that you can lose all your phosphate in your kidneys. You get hypophosphatemia if you have too much FGF23. But nature makes it in such a way that there is a competition between the addition of an O-glycosylation site here or a phosphorylation site that we're going to discuss. Once the phosphorylation is added, now it can be cleaned and you can save from losing your phosphate and your bones develop normal. So you can see that not only these modifications can change the shape of the protein, but they can make a decision on whether the fate of the protein, what's going to happen to it. Is it going to stay as it is? Is it going to change size? And it's modification that does that. We take, for example, a protein that we're going to talk about, which is the very close to the transmembrane domain. It's the type 1 to remind you of this protein. People didn't understand, why do we need those? Well, you can now express the protein in a cell that doesn't make the O-glycosylation. What do you get? It's in the media. The enzymes start chewing it up, and you don't have a receptor. So O-glycosylation is also a stabilization of protein to prevent from it being degraded to earth. And so on. I'm not going to go. This is the O-glycosylation is very important for the development of Alzheimer's disease, in the case of the A-beta formation. So it does a lot of roles. And if you want to read about it, I think Henry Clausen is the one who's done the cloning of the 20 enzymes involved here. Another modification that Dr. Ferron is here and is very interested in in the industry. It's called the GLUT. It's a vitamin K dependent gamma carboxylate. What it is? You're basically transforming a glutamic acid into a double carboxylate. It's called the GLUT. Right? And that's very important in blood coagulation, and also in bone formation. Right? So again, not only are you going to be able to transform a serine or a threonine in an O-glycosylation, asparagine by N-glycosylation, but you can now transform specific glutamic acids into GLUTs. What does it do? It adds 2 negative charges as opposed to 1. So you've changed the charge of the first. Very important for bone formation, very important for blood coagulation. If you hurt yourself and you cut yourself, if you do not have vitamin, or you add warfarin to the blocks, basically, you'll bleed. Okay? You have problems. You cannot coagulate your blood. So you need that modification. Another one is called acylation. Cysteines are strange amino acids. Usually they tend to like to make disulfide bridges. But if a cysteine is in the cytosol, like the cytosolic tail of the transmembrane protein, it is very reactive, cysteine. It doesn't stay alone. Okay? So usually the cells make sure that you cover it by what is known as a palmitoylation, 16 carbons that you're adding to it. What would that do? You're adding CH2 16 times to the system. It will make it highly hydrophobic. It will love to stick to membranes. So anytime you see a protein that has a 15 palmitylation, you can bet on it. It's a membrane, very strongly attached membrane protein. It's one way to anchor it very tightly to the membrane. You can play the game of depalmitylation and palmitylation to move it from one compartment to another. So again, this modification gives you diversity because it allows you to move from compartments by just changing or not having this modification. It's a reversible one, as I just said. An interesting one was discovered many, many years ago and finally solved in Texas a few years ago. It's called serine octolylation. Basically, it's a serine again. It's not N-glycosylation, the O-glycosylation. It's not phosphorylation, but it is octolylation. It means adding 8 CH2 groups. What is that? This was the serine octolylation is found in peptides. Usually, some peptides have in your gut. You will not be able to digest your food if you do not have the serine octolylation. Believe it or not. In fact, you will not be able to eat because you will not be hungry. Some companies are actually now making inhibitors of this modification to prevent overly obese people from eating. Because that's why would it give you so much appetite. It turns out there is a hormone called ghrelin in your stomach that is a signal that tells you eat, hunger, eat. Okay? How does ghrelin work? So ghrelin is made as a precursor, okay? And it has to be serine octolylated, as you can see this. CH3, 8 carbons added to this serine. Once it's added to this serine, it will now tell your body, eat. Okay? Or it gives you a loss of satiety. You have to eat more. So octolylation is a signal. It's not used very often, but it's a very typical case where it affects the activity of ghrelin, which will then have a number of functions. It will affect growth hormone, will affect leptin, will affect a lot of things to be able to give you this sensation of hunger. Now, what about, we have 20 amino acids. So you can imagine that if you were God and you were thinking, how am I going to add all these modifications to my proteins? I can use any of these amino acids and modify if I can make them reactive enough. Tyrosine is another one. So usually tyrosine is modified by sulfation. You can add a sulfate group. What would sulfate group do? It will add a negative charge. Once you add a negative charge, you can now modify the function of the protein. Or you can add phosphorylation. In fact, 75% of the protein in our brain, okay, are phosphorylated on serine and threonine. So much of your listening to me today, okay, of your memory of what you're looking at me, in large part, is due to your ability to phosphorylate proteins in your brain. We're going to talk about. Here's the kind of protein that we discovered, and I'll talk about it at the end of the talk. It's a protein that is a major player in regulating your cholesterol. It's now clinical trial. The clinical trial has been ended, and it's on the market right now, the treatment of this protein. But this is a protein that is decorated also. It has sugars and glycosylation sites here, and X, S, S or T. It has two phosphorylation sites, and it has a sulfation site on its axis. It has all these modifications. You can play the game. Okay? So I want to show you this phosphorylation site, because it took 130 years. In 1880, all of Hammerstedt in Sweden, okay, was the first to look at milk, the mother's milk, when she feeds a baby. And he says, that's funny, this milk, has some phosphate. That time the chemists had began to know what phosphate is all about. He said, milk has phosphate. And he isolated a protein from milk, one of the major proteins, called casein. That's what you see today. He did a lot of casein. And he was able to isolate it. This is 1880. No, this is early, 1883. He was able to isolate the milk and found out that actually phosphate is attached to the protein. So that was the first evidence that there exists phosphorylated protein. No one understood how and why. But it's there. Casein is very phosphorylated. In fact, milk, many proteins in milk are highly phosphorylated because you want them to be very solid. Phosphate means negative charge. Okay? And it took a number of years. People didn't understand what it was. Was it one enzyme that does this? How many enzymes? And, kinases, there's about 700-800 kinases in the cytosol of the cell. But this is not a cytosolic protein. Casein is a secretory one. It has a single peptide, it can go to human, and so on. It is not in the cytosol. So what is the kinase responsible for the phosphorylation of casein? It was just, and the person who done it is giving a talk here on Monday, whenever he comes. It's called Vincent Tagliavacci from Jack Dixon's lab. He was able in 2012, 130 years later, to finally identify the kinase. And it turns out that there's only one kinase. There's 700 kinases in the cytosol, but only one kinase in the secretory pathway. So, how did he do it? He did a lot of PCRs, and he was able to identify a family of kinases, but only one of them is in the secretory pathway, and it's phosphorylates serine. Another one that he identified phosphorylates sugar, because phosphorylate sugars, glycosaminoglycan. But to phosphorylate the serine, there's only one. It's called family C, 20C. Interestingly, he just discovered that it's activated by a lipid called sphingosine. Depending on your diet, you can have more or less sphingosine. And you can have more or less, this sphingosine activates 130-fold the activity of this kinase. So that means your diet will affect how you phosphorylate certain proteins or not. People didn't realize that. You can modify that. It turns out that phosphorylation is protected, so you're better off to have phosphorylated proteins than dephosphorylate. And it's not reversible, different from the cytosol, which have phosphatases. There is no phosphatase in the secretory pathway. So once you add the phosphate, forever. And then he discovered another kinase that phosphorylated privately. So what do we learn from this protein? It's a single kinase, it's not too big, as you can see about 400 amino acids. It is stuck in the in the lumen of the Golgi. So first thing, we knew for many years that the phosphorylation of proteins occurs in the Golgi apparatus. We didn't understand why. Why the Golgi? I think we're beginning to understand. Because it gets activated in the Golgi itself. And it's very, very specific, this enzyme. And you better look at your protein. If you're working with proteins, check for a single motif. It's a very simple one. Serine X glutamic acid or a phosphorylated serine. Whenever you have a serine X negative charge like glutamic acid, you will get phosphorylated. So this is just emerging. Very new, I'm telling you. But last year it's beginning to really explode. Now, turns out that it's not so simple, but there is another motif, SQ, XXED, or ED. That's the third one that's more rare. The common one is this one. All right. Now, you can now take any cell, in this case it's HG2 cell, isolate the proteins, and then modify, see which one of them are phosphorylated. This is a HG2 cell, it's a liver cell, and you can do mass spectrometry to identify the phosphorylated proteins. You can see that many proteins. Many that nobody even thought about. Okay? So think about it. This is a modification that will modify deeply your ability to have a certain function, and it can be modified by diet, by sphingosine. Very important. Right. So here's the protein, it gets translated, as I told you in the lumen. Here is the kinase. And they both meet in the Golgi itself. The kinase is activated. The kinase is activated. Here's the protein, and the kinase is in red. Once the kinase is activated, it will need ATP that will have to go in, because it requires ATP and magnesium, and it will phosphorylate the protein. And it gets that. So, remember, this kind of phosphorylation is not in the ER like the asparagine. Glycosylation, as I told you, that occurs in the endoplasmic reticulum. We're now into the Golgi. We're moving in. Now we're moving in even further. We're going to go now to the last lamelate of the Golgi. This is where the game starts. This is where you get the biggest diversity. This is where a protein can give you up to 60 different fragments. Imagine, one gene will give you 60 different proteins. Right? That's quite a bit. That is done by an irreversible modification called the phosphorylated cleavage. Now, if you look at the genome that we have, the enzymes that are in called proteases now, okay, they only represent about 2.8% of the total genome that we have. So 2.8% of our genome is involved in cleavage, cutting. Of these, 90% degrade. Their function is not to actually generate a new function, is to bring it back to amino acids. 10% make the big job of actually transforming a protein into a different form. Now there are 5 different kinds of proteins. And they are divided based on the amino acid that is critical for their activity. For example, an aspartic protease that is involved in Alzheimer's disease, for example, everybody's been using this as an inhibitor. HIV protease that is now being used as an antiretroviral drug. These are aspartate proteases. They require specific aspartic acid for the activity of the enzyme. The drug that they're targeting the aspartic acid. Cysteine proteases are usually digestive proteins, but they also can do other stuff. Metalloproteases are notorious. This is what causes cancer metastasis. Okay? All the cells that have to migrate out and go in different places will require metalloproteases to open the field for them to start moving outside of the cell, outside of the extracellular matrix. Serine proteases are the most famous one, and these are the ones we're going to concentrate on. And finally, the threonine proteases are what is known as the proteasome. After we're going to talk about proteases in the cytosol. Proteasome is the garbage collector. It's the one that cleans up the proteins of the cytosol. These are threonine proteases. Now, what are these proteins? Well, we have what is known as the C-terminal processing modification. So, when a protein is cut, here's the protein that's going to be cut. Okay? Specific site. It doesn't matter for example. We're cutting a peptide bond, right? Here's the tyrosine sulfation. Here's the acylation of the N-terminus, and so on. Once it's cut, if there is a dibasic residue or a basic residue at the C-terminus, you have an enzyme called the carboxypeptidase that will clear it out. So that's why when you isolate a hormone that you're thinking that is doing your job, you never find the terminal oxygen residue because they're removed by the carboxypeptidase. If ever there is a glycine just before this dibasic residue, it will be amidated by an enzyme called the amidation enzyme. It's a combination of 2 enzymes. Now, amidation enzyme, if you try to knock out this enzyme, you die. You have no baby, no fetus without the amidation enzyme. Clearly, many of our hormones require amidation. Vasopressin, for example, for control of your blood pressure. Oxytocin, for a woman who is getting a baby and needs to deliver the baby, requires an amidation. Many of the hormones that we have in our body have amidated C-terminus because the receptors recognize this function. Today we're going to concentrate on actual cleavage. What are the enzymes that cleave a protein and transform it into different? So you can get an intracellular or cell surface cleavage, or you can get an extracellular. Those metalloproteases are notorious to allow cancer cells to move. If you cut yourself, for example, and you get blood, you need to make clots. Okay? So you have 12 different enzymes in your blood that will make sure that you will clot your blood so that you don't bleed to death. Okay? This is called extracellular cleavage. I'm not going to be talking about it. It's another thing. We're going to be talking about the intracellular cleavage. Once you go into the intracellular cleavage, you can get serine protein, usually they are serine protein, or metalloproteins. So let's talk about the intracellular cleavage. We now begin to bring the term, the protein convertases. I worked on this 3 years to be able to identify the enzymes I'm going to talk about. It was very difficult. Very tough enzyme to isolate, very resilient to purification. We had to use all types of methodologies to identify. But they're there. And it's like this guy who's trying to, stay under his umbrella, and some smart Alex decided to cut a hole in the umbrella, and as he stayed, supposedly he was in this shape, he gets tanned just the same size as the cut. The point I want to make here is that the convertases that we talk about are very, very specific. They don't cut anywhere. They cut at a specific position to create a new function. That's the game here. We, out of one protein, create more than one function. That's the point. Right. And I want you to learn a nomenclature. In the literature, it's important because you'll see many of these papers. Imagine that this surface here is an enzyme. And imagine that this peptide here is entering inside the catalytic pocket of the enzyme. Okay? Because that's how the enzyme is going to cut. It's going to have to accept the protein or the peptide in its pocket and cut it. Here's the cleavage site, right there. So we define that the cleavage site and terminus to the cleavage site is called the P1, P2, P3, P4 position. Here, P1, P2, P3, P4 position. And what's after the cleavage site? C-terminus is defined. Everybody uses this nomenclature. P1 prime, P2 prime, P3 prime. So when you tell you that this enzyme is specific for an arginine residue, it is specific for a P1 arginine. So that's the specificity of the cleavage. Right. So what happens when you do it? Here we have a protein, doesn't matter which one. It's a soluble protein, you see, or you can have a membrane-bound protein here, 1 time or 2 times, or whatever you want. It has the sigma peptide, or it has this transmembrane domain, it has the visa to get it. It's going into the ER. Right. Once it goes in, it will add the sugars and so on, it will move to the Golgi. Once it reaches the Golgi, now a decision has to be made. Are we going to allow this protein to get out as such, or are we going to cut it in different pieces? Are we going to be the tailor that's going to make a dress out of this polar protein that doesn't have any shape and transform it into actually very active fragments? So there are these motifs here that we're going to talk about that the convertases will require a recognition motif to cut them. Usually we thought, until about 2-3 years ago, that this cutting will now generate a piece here out of the active protein. We usually thought that this was turn on. In other words, we're creating a sequence, we're creating a structure, we're getting a gain of function. But we recently showed that this kind of cleavage can actually be a loss of function. It can actually transform, like we saw in FGF23. If it's cleaved, it's lost its function. Okay? We can get it now more and more we begin to see that. So, cleavage is a way to either generate a new function or lose it. And it's up to the cell to make a decision which direction to go in. So this is the kind of timetable from the Mendelian days until we are now. It took many years until the yeast was the first source of the solution to the problem. What are the enzymes that cut the protein in the 6-30 phospholipid? The yeast is a much simpler organism, a single cell, okay? But it's different, like similar to us, it has to mate. We have to mate to get children and so on. But the yeast too, the same thing too. They need a hormone called alpha mating factor. For them to mate, the haploids will mate and make a diploid. And so the question is, what activates this hormone? How do they mate? So you can use the yeast mutants to isolate mutants that don't mate. And then ask the question, what happened? What are the genes that are modified? So Jeremy Thorner in 1984 was able to identify the first processing enzyme in it. He calls it Cas2 protease. But really, the human equivalent was not discovered, but many years ago. By John Hutton, who was interested in a tumor. He had a lady who came to the clinic with a huge tumor of the pancreas. going to die. And she had to, they offered him to cut out the tumor. And he got the tumor. So he figured that this tumor must be producing a lot of insulin. Okay. And he was able to extract the tumor, which has so much insulin now. He said there must be the enzyme that activates insulin out of this. And he in fact isolated biochemically two enzymes, he called it type 1 and type 2, from this tumor. And he said that for making insulin, you need two enzymes, but he had not cloned them. He just knew that there was two activities. But it was the first thought that to make insulin, it doesn't take one enzyme, it takes two. This is where we came in, and we started isolating the whole family of convertases. It's a long story, as you can see, it took 13 years. But from 1990 all the way now, to 2002 and 2003, we isolated the 9 enzymes in this family. Okay? They turned out to be similar to each other, the first 7, and I want you to remember that. And then the other 2, we will see, this is the one that is in clinic right now. Okay? So, today what I'd like you to get an idea, you know that there are 9 enzymes in the secretory pathway that will shape your proteins. I want you to understand that each enzyme has its own function. Okay, they're not the same. So when you come out of here, I'd like you to remember that there are 9 of them, and depending on which one you're going to use, you're going to create different kinds of proteins. I want you to learn what do we know about them so far. So here they are. They turned out all of them to be serine proteases of the subthalide enzyme. Now this was unexpected. I was skiing with the best enzymologists in the world in Austria, and he was telling me, no way, it's going to be serine protease related to trypsin or chymotrypsin because bacteria are too far away. It cannot be bacteria, but it was wrong. It turned out that this is a family of serine protease, has a serine of the active site, and they are much more related to bacteria than they are to the blood coagulation sites, which are serine proteases also. So it's a very ancient program. We estimate around 600 million years ago at least they appeared. 600 to 800 million years. The dinosaurs had it, plants have done a lot of things. So, what's so special about these enzymes? Well, all of them have a single peptide. This is why they end up in secretory pathway. I told you in the beginning. So that's second one. Then they have a strange domain in red here. It's called the prodomain. If you remove the prodomain, they cannot get out of the endoplasmic reticulum. Why? Well, they cannot get out of the endoplasmic reticulum because they cannot fold. They don't get the right shape. And there is this quality control protein that says, no way, you don't get a visa to get out of the cell. So they need help to fold, to get the right fold. And help is done by this red domain. This is called the intramolecular chaperone. It chaperones out the protein to adopt the right conformation. And once it adopts the right conformation, the quality control system says, okay, I'll let you go. You go. Take it out. Most of the enzyme you will see will get rid of this red part, so that because it turns out that this little red part will bind to the catalytic subunit of the enzyme and inhibit it. As long as the prodomain is there, these enzymes are not active. So they have to find a way for them to become active later on. So it's one way to regulate it. And then they have different domains, each one of them, that tells them where to go inside the cell. So if you have a key that can open up many doors, that's great, but how do you know which door you have to go in? You need to get an address. You need to know where you're going. And so you have domains here that tells it interact with protein A, B, C, D, and that will send you to granules, it will send you to the cell surface, it will send you to lysosomes, it will send you to endosomes, it will keep you in the Golgi, it will allow you to cycle back and forth from different organelles, and so on. It's all defined by these domains. Of these 7 first convertases, we discovered that the 7 first cleave proteins only at basic residues. Usually it's an arginine. It must be preceded by another arginine or lysine 1 at the P2, P4, or P6 position. So there must be 2 basic residues for a convertase to function. It cannot function by a single basic residue. Whereas thrombin, for example, only requires 1. Then there are these 2 strange enzymes that when we discovered them, we didn't understand what was going on. They are very similar, as you can see, in terms of structure, except they don't cut at basic residues. This one cuts at a non-basic residue, but it requires a P4 arginine. This one, you will see, is a major player that regulates the synthesis of acids, cholesterol. If you do not have SCI1, you don't have the brain, right? We've created animals, chicken and so on without this. So you just don't have a brain, right? It's a major player in defining how your body is going to die. You see some examples. And finally, you have the last one, which is a weird one. It cleaves itself once, because all of these enzymes, I can tell you, autocatalytically cleave themselves in the ER. So in other words, the removal of this red part is done by the enzyme itself. It recognizes its own motif within. And the only one is this one that cleaves itself but never gets rid of its protein. What does that mean? It means it will never become an enzyme afterwards. Why would God create an enzyme to kill it? You'll understand as we move on. So, I want you to remember this. So, all enzymes, all these convertases are synthesized as precursors. They are inactive because the prodomain is inside the catalytic subunit. It allows it to get the right shape. That's it. That's the chaperone. That's fine. Okay? But then it has to get rid of it. It has to be able to get rid of it to become an active enzyme. So it gets the visa, you're cut, you can get out. It gets out. It goes to the Golgi or it goes to the granules or whatever. It's still dead. The enzyme cannot function. Until it reaches the right compartment with the right concentration of calcium, pH, and so on, it will now usually cut itself a second time. Once it becomes active, it can hit any other substance. And you see some of the examples. So you can see that in order to go from an enzyme that is started to an active enzyme, there is some regulatory steps to define where the active enzyme is going to be. So we've done a lot of cell biology here, a lot of electron microscopy and so on, to identify where these enzymes are. But it turns out that actually they're all over the place. In simplest one are called the first two enzymes identified called PC1 and PC2, proprotein convertase 1 and proprotein convertase 2. They are the ones who make all the hormones of your body. If you have a memory problem, or if you have pain, or if you have insulin sensitivity, glucagon, whatever, all the hormones of your body are made by these two enzymes that cooperate. The other five, they go into the last lamellae of the Golgi, and they can go to the cell surface and back. They can do that. Viruses and bacteria will love that. Because they say, why should I synthesize an enzyme to activate my surface glycoprotein? I just have to exploit the ability of the cell to bring me this enzyme at the cell surface, get cut, and infect. Exactly what the viruses do. You've heard about the Ebola virus, you've heard about the HIV virus. They all use the same program. They exploit the ability of the cell to cut them at the cell surface together. Two enzymes called PACE1 and PC5 can get out of the cell surface. They're soluble, as you can see, but they're recaptured again by heparan sulfate protein glycan. These are very negatively charged. These are extremely important for the body to decide whether you have how many ribs. They decide its direction. It's really polar. This is the interactions with specific heparan sulfate protein that will polarize them along the axis of your body. So, the distribution of these enzymes is better. The simplest one is CC4. It turns out to be only in the testes and in the ovaries, and it's a major one for reproduction. If you do not have PC4, you are infertile. Simple. So we try to, unfortunately, our society is more for female than we try to use this to make a male contraceptive. It's very easy. If you block PC4, your sperms cannot activate if you want to penetrate the ovum. But the companies don't want that. But we'll find a way to somehow commercialize this. Because this is a major player to allow, it's a germinal cell, that allows basically the sperm to get into the ovum. Okay? That's the simplest one. More complicated ones, I told you the PC1 and PC2, they are all endocrine granules, or these big granules, I told you that store hormones. Furin is everywhere, we'll come back to it. PC5 is widespread, and secreted. PACE4 also. PC7 you will see, it's not secreted, it's ubiquitous, but it's membrane bound. C1 is also in the Golgi. It never gets to the cell surface. And finally, PCSK9 is only in the liver, small intestine, and kidney, and that's what we're going to talk about for the clinical application. We're going to come to that one. What do they do? What kind of substrates do they cut? It took a number of years, a number of people around the world, once these were identified, to say, hey, spike protein cut. Where is it cut? What does it do? So you can see that many of the hormones are cut by PC1 and PC2, the glucagon, the alpha MSH, the GLP1, ACTH. surfactants, receptors, adhesion molecules, metalloproteases, even viral vector proteins like HIV. I told you, use it to get it. In fact, it's now in clinic for that. Okay. And bacterial toxins such as the anthrax toxin. You heard about the White House when they got an anthrax letter, it was full of anthrax, and they began to give them some of these urine inhibitors just in case, to block the entry of the anthrax. PC7, we'll come back to this. It affects a specific protease. SCI1, we'll talk about it, and also PCSK9. These are the clinical trials. That's for you to know. If you're interested, this is the Clinical Research Institute. We're very much interested in seeing what we've learned of these enzymes can you actually find in clinical application. These are all over the world, the clinical application of each one of these enzymes. The most advanced ones are PCSK9, and we'll talk about it at the end. For both hypercholesterolemia, if you have too much cholesterol and you're resistant.
It will freeze. You can see the animal going like this. It freezes. Scared that it's going to be sent. The knockout doesn't care. See, the knockout is not scared at all. So already that was the first indication that the loss of PC7, you're not anxious. You lose anxiety. That's an important disease in you. Okay? You can see that it just doesn't freeze. And what you do, you do another test. You take the same animal and now put it in a different box. Never seen this box before. Change the color of the walls. So nothing happens. And all you do is a few minutes later, put the tone. Remember that there is an association of the tone with a zap. Okay? The normal mouse will freeze very fast, but not the knockout. So we interpret this from many other tests, that the animals are cool. We call it the cool mouse. It's just not scared. Okay? It's lost anxiety. And if this was due to BDNF, we can give them back this VHF drug. And if it's true, it should revert the phenotype. And it does. Completely reverts the phenotype. So it's a lack of BDNF. So it's reversed completely with the hydroxychloroquine. So we now know that PC7 is a major player in anxiety, and we are now beginning to get drugs for, you've seen our soldiers that come back from Afghanistan and so on, or people who have a lot of anxiety disorders, and we're beginning to think that this is going to be an interesting drug. It's difficult, you have to cross the blood-brain barrier to get the drugs through, but we think it's going to be a very, very new way of controlling emotions, or a very unregulated emotions. SCI1 number 8 is another enzyme that is now found early on in the secretory pathway, way before the fuel, as well as PCSK9. These are the two enzymes that play a role in cholesterol metabolism. Okay? Now, SCI1 itself, it's also autokinetically cleaved by its prodomain at 3 positions, but now the cleavage site is no longer at the basic residue. It's an arginine at P4, an aliphatic at P2, and then a small amino acids. Why is that important? It's important because for the synthesis of cholesterol and fatty acid, there are transcription factors, membrane bound, that need to be cleaved to make cholesterol and fatty acids. Without SCI, you die. The embryo dies at embryonic day 2. There is no baby. There is no blastocyst. Clearly, this is a major, major player. Okay? And we are now discovering it has other functions. It can actually activate the additional phosphate on mannose that allows protein to go to the lysosome, the phosphomannose binding. And viruses, such as the Lassa virus and the Crimean Congo hemorrhagic fever virus, these are hemorrhagic viruses. There was in Namibia, there was a mine that was mining, if you want, some very, very important jewels and diamonds, basically, and the diamonds were stolen, you know? And so the guy who owned this mine says, what's going on? Why are my diamonds being stolen, you know? So he sent some spies to find out what's going on. But he always found that when the diamonds were stolen, there were some people, the thieves were dead at the entrance of the mine. So he didn't figure out what was going on. So he found out that it actually, the mine was infested by bats. Okay? Bats were carrying the Crimean Congo hemorrhagic fever virus. Okay? And that's a killer. It causes all your blood vessels to explode, and you die within a day or two. Okay? So these guys were trying to steal the llamas, but they got bitten by the bats. You know? And that's how the virus got transmitted. So clearly this is an application for this kind of diseases where there is absolutely no vaccine, no treatment whatsoever. And in Africa, it's a really horrible disease. So, SCI1 is necessary to deactivating of the two major transcription factors, very important for the synthesis of cholesterol and triglycerides. And for those of you who have too much cholesterol in your body, you get something called statins. Okay? And statins are inhibitors of HMG-CoA reductase. This is how you diminish your cholesterol synthesis. Okay? So SCI1 is about that. And as I told you, it is very important to activate this complex protein that is important to add mannose, phosphate of mannoses in glycosylated proteins. Without this, you don't go to lysosomes. Simple. So here's a goal. An enzyme that is necessary to tag proteins to go to lysosomes by activating a kinase. Now, as I told you, there is no baby, there's just no epiblast formation. They die at embryonic day 3 or 4. So how do you do with something like that? That's another point. If you have an enzyme that the animal is dead, what do you do to find out what is the function? That's complex genetics. We do it in the lab. And that's called tissue-specific knockout. It's just to make it fun. We've never seen a mouse with a blue eye. But you can actually remove the gene in specific tissues, okay? And ask the question whether you can get a phenotype. We've done that. So SCI1, if you remove it specifically in the liver, your cholesterol and triglyceride drop by at least 50%. Okay, this is an 80% knockout, 100% you die. But 80% are still alive, and the cholesterol and fatty acid is really down by 50%. So clearly it is playing a role in the synthesis of cholesterol and fatty acids. So we just got a paper out on this is SCI that you can see it all over the mouse. It's expressed in many, many tissues. Okay? And now we ask ourselves the question, what happens if we remove it just in what is called a CRE promoter, the collagen promoter, collagen 1 CRE, okay, in chondrocytes? What happens? Well, we get a very strange animal. It has a kinky tail. The tail turns like this, as you can see, much shorter. And you get vertebrae that are fused. Okay? This has nothing to do with cholesterol or fatty acids. This has to do with another transcription factor called CREP that SCI activates to be able to get your spine. Your spine to have all the different parts. It requires SCI to be able to separate it out in space. So basically the extracellular matrix of the bone is extremely sensitive to SCI1. Let me finish with the PCSK9, which is the one that is the most exciting right now. It's the last baby of the family, was discovered in 2002, and being in this institute and being close to Jean Davignon, the guru, if you want, in Quebec, for the lipids, he was my mentor, and I was told for many years that the biggest diseases that used to affect our society in the 1900s have decreased a lot. And really the major killers right now are cancer and heart disease. These are the major killers right now. So I want to show you that if you look at the bloodstream, what do you see? You see that you have in the bloodstream a number of lipoproteins, including LDLs and HDLs. The LDL are the green one. Okay, this is the bad cholesterol. But there's also a good cholesterol called the HDL, which distributes the cholesterol in different tissues and picks up the excess and sends it to the liver. So there's always a fight between the HDL and the LDL, low density lipoprotein and high density lipoprotein. So the high density lipoprotein is more complex, but it picks up the bad cholesterol and sends it to the liver for degradation. It is the LDL that is the real problem. People have a lot of LDL get heart attacks. And this is serious in Quebec. 30% of our population have high levels of LDL. This has to do with, well, now we are more mixed society, but it's a founder effect. It comes from France and so on. And so when we discovered PCSK9, we asked ourselves the question, well, it's an enzyme, right? We always do this for all the convertases. What does it do? Well, it autocatalytically cleaved itself, here it is again, but something funny happened along the way. It never gets rid of its prodomain because it's secreted with its prodomain. Wow. That according to what I told you, if it's secreted with the prodomain, it cannot act as an enzyme anymore because the prodomain is an inhibitor. So why would you create an enzyme to kill it? That is the procyclin. But it also got killed by another enzyme called furin by getting cut. So it's a double killer. Why would you do that? Well, if we look at the way we understood the way PCSK9 is, it gets out of the ER, the same story again, as all the convertases move to the Golgi, all the enzymes get rid of the prodomain except PCSK9, which is secreted with its prodomain, and it's inactive. Remember that. It's no longer active. It only acts as an enzyme once to cut itself, to get out of the ER. Crystallize it, look at its crystal structure. Crystal structure told us what's going on. It told us that the prodomain, this N-terminal inhibitor, is really stuck in the catalytic subunit of the enzyme. And it's so stuck that you cannot pull it out. It's so tight in that cavity, you cannot pull it out. It's the tightest cavity of all the proteins. So the structure of the PCSK9 told us there's something about this enzyme that shouldn't act as an enzyme anymore. But what is that? So for that, we were lost for a number of months. We really didn't know what to do with it. So we said, well, listen, what do we know about this enzyme? Well, we know that it is rich in the liver. You can see in the liver, very rich in the enzyme. So that was the first clue. It's also on chromosome 1p32. That was the second human chromosome 1p32. So what do we know about this? So you go into Google. Everybody goes to Google, right? And you say, who's working on chromosome 1p32? Wow. Turns out that there is a French team been working on this chromosome 1p32 for 5 years. Why did they do that? Well, they work on this part of the chromosome because they were looking for a third locus of hypercholesterolemia. Wow, liver, I called them up. I didn't know them. They didn't know me. And within a few hours, half an hour of speaking, we got into collaboration. I asked them, listen, maybe what you're looking for is this enzyme. So, okay, if it is the truth, we'll buy you a bottle of champagne. Okay. They bought me 3 bottles of champagne. It turned out to be the right one. Okay? We are friends ever since. And these are the kind of patients we see. Okay? These are patients who have extensions in cholesterol. These are xanthomas and xanthelasmas. Okay? Look at yourself in the mirror. Hope you don't have this. Okay? If you have this, you have a problem of cholesterol. Okay? And a very famous person had this problem, but when she was acting, nobody noticed it. And Leonardo da Vinci painted her, the Mona Lisa. Turned out that Mona Lisa died at the age of 37 of heart attack. Why? Well, look at it carefully. She has a grand xanthelasma. The painter saw it, but he didn't know. This was 16th century. He didn't know about cholesterol and that, And I went to the Louvre, all these places with Italians, and Mona Lisa doesn't have it. So no, it's there. Okay? Well, so many people didn't even notice that she was actually hypercholesterol. So cholesterol is an interesting story. It's really started with Carl Muller in 1939 in Sweden. Okay? He's the first one to realize that cholesterol is, LDL cholesterol is actually dangerous for your body. And this really came from Khachaturian in Lebanon, who was the first to show that hypercholesterolemia is a genetic disease. It's a specific genes, when they're mutated, cause this disease. In fact, it turns out that in the Middle East, in Lebanon, there's a lot of this hypercholesterolemia. So Khachaturian has been the first in 64. In 74, Goldstein Brown discovered what is the gene that is responsible for this disease, and it turned out to be the receptor of LDL, the bad cholesterol. They got a Nobel Prize for it. Okay? And again, Iñárritu discovered the ligand of that receptor. It turned out to be ApoB from the LDL, the protein that surrounds the LDL. So if you have mutations in the protein that surrounds the LDL or in its receptor, you get the same thing, hypercholesterol. And until we discovered our gene, we thought our story is over. People have this type of problem, you give them statins and you diminish cholesterol synthesis. Hypercholesterolemia is a deadly disease. It kills at least 30 million people every year. Okay? And its frequency is around 1 to 200, 1 to 80. In Saguenay-Lac-Saint-Jean here, 1 to 45. Lots of people have this disease. lots of problems, and you have to be treated very early. The children have to be treated as early as the age of 8. Okay? So, patients are not able to reach sometimes the LDL target, even under statins. They cannot go down. And so they will get a heart attack if they're not looking. So people were looking for a new treatment for hypercholesterolemia, which is a deadly killer. It takes years. That's a killer. Until we came up with this story, here's Mariana B. Fadel. She's from Lebanon, and Dr. Catherine Boileau. She's from La Salpêtrière in Paris. And they were looking for this family from Nantes and Bordeaux for years, 5 years, trying to find out what is the gene that is involved in this disease. Until we told them it could be PCSK9, and I finally discovered that indeed all the families had mutations, single point mutation, phenylalanine to leucine, or a serine to an arginine in PCSK9. So that was a single point mutation. One amino acid change can increase your cholesterol by 3-fold. That's a lot. Now, once this was published, it's the Olympics. You know, everybody jumped on the bandwagon and said, hey, I'll look at that, what about my gene? I'm going to check my patient and so on. Until the Mormons in the United States, and these are actually an Anglo-Saxon mutation, discovered that in the Mormons in Utah, there's a lot of children who die of heart attacks at the age of 3, 5. Okay? Well, it turned out to be a very dangerous mutation. It's just transpartic to the tyrosine, which increases the affinity of PCSK9 to the receptor LDLR by 20-fold. So it's a gain of function, huge gain of function. In fact, it was discovered in Norway, and all you can see in Norway here, and also in different parts of the world. On the other side, it was discovered that you can have different other mutations that will result in loss of function. And these people would have very low cholesterol. In fact, we discovered 3 people around the world that had so low cholesterol it was never reached by anybody by any drug. 0.4 millimole. This was a woman in Texas. She's a double heterozygote. Another woman in Zimbabwe. She was being tested for HIV. She turned out to be a homozygote mutant of PCSK9 that kills its function. And the third person was diabetes, unfortunately, in Nantes. He also has a double heterozygote mutation. Basically, it says that you can live without PCSK9. Not only can you live, but your cholesterol is very low. This woman here is an aerobic teacher. Okay? She's a comparatively good athlete, even though she weighs 170 pounds and measures 5 feet. For me, she's fat, but I was told that this is the norm in Texas. So that's what it is. But it turns out that at least you can live. A human can live without PCSK9 and have a very low cholesterol. Okay? So, what we're saying is that there are gain of function mutations that are dangerous, will give you high cholesterol, and there will be loss of function mutations that will give you low cholesterol. So then, companies said, oh my God, if that's the case, let's try and kill PCSK9 so that we can get low cholesterol. So we now understood why it's working, because basically it targets the LDL receptor to lysosomes for degradation. So the more PCSK9 you have, the less LDL receptor you have, and hence more LDL circulating because you cannot clear it. Okay? And you get hypercholesterolemia. Okay? So what was thought to be so simple before is that the LDL binds the LDL receptor, goes into the endosomes, pH drops, separates out, and you distribute cholesterol in the cell, and the LDL receptor comes back to pick up new LDL. Is no longer true, because there is a new player in the game, and that's PCSK9. PCSK9 now, once it binds the LDL receptor outside, it sends it to lysosome and it doesn't recycle. So that means the more PCSK9 you have, or the more active PCSK9 you have, this is the Mormon mutation. You get a lot of LDL, you get heart attack. This was co-crystallized with the PCSK9, and we now know that it is the PCSK9 binds a very specific domain of the LDL receptor called the EGFR. Right. Now, can we reproduce this in animals? We created mice without PCSK9. Look at the amount of LDL receptor at the cell surface. This is a normal mouse. We remove PCSK9, huge amount of LDL receptor at the cell surface because it's not getting degraded. And what happens? The cholesterol drops. By 80%, we lose the LDL. These mice live long because they're hypocholesterolemia. So that was nice and dandy. In my lab, there is 14 women and 3 men. So we're surrounded by people saying, why do you work always on the male, work on the female, and see if there is a difference. We did that. To our surprise, we found there's a big difference between the male and the female. That's important in terms of clinic. While the male has a huge increase in the LDL receptor at the cell surface if you use PCSK9, because it doesn't get degraded, the female, you don't see it. And yet, the amount of LDL receptor by Western blot is the same. So the females handle the cycling, the movement of the LDL receptor in the cell very different from the male. The end result is the same, but the trafficking is completely different. And so we wanted to know why, and we've overrectomized our mice, our female mice. And when we overrectomize a female, it becomes like a male. It now expresses the LDL receptor back at the cell surface in large amounts. And if you give them estrogens, it comes back. It goes back to ovary. It comes back to like the female. So clearly estrogens are modifying the way LDL receptor is cycled to the cell surface in the absence of PCSK. Very important in terms of treatment. So plasma analysis realized that people have a correct correction between the LDL receptor, cholesterol, and PCSK9. So the objective then is diminish PCSK9. We do a large study here, 3,500 people, and asked to serve the question in this room. There may be some of you who have lost one of your allele because you're lucky from one of your parents, let's say. This is called a heterozygote, if you want. And it's not so uncommon, by the way. And we found that this was 12 patients who lost one copy of PCSK9. And if you look at over 15 years, the ability to get a heart attack is decreased by 50%. And 88% if they're heterozygote, 88% went down on any cardiovascular event. So losing one allele of PCSK9, decreasing 50% the activity of PCSK9 protects you against heart attacks over a lifetime. So the objective then is give them statins to reduce the cholesterol production and give them an inhibitor of PCSK9 to increase the amount of LDL receptor in the cells. That's what the conference talked about. So did we. And so this is what's on the market right now. It just got accepted about 2 months ago. There are 3 companies now selling this antibody. It's a monoclonal antibody. You inject subcutaneously the antibody every 2 weeks. There are small injectors, not like for diabetes. It will bind the PCSK9 circulating, prevent it from interacting with the LDLR, and increase the amount of LDLR on the cell surface. This way, you pick up the bad cholesterol. Does it work? Does it ever work? This is now the first, what we call outcomes. When you do any clinical trials, it's nice to say, yes, I've diminished cholesterol, but what does it do to the lifespan of a person? What does it do to its ability to get a heart attack? This is called outcome. It takes many more years to be able to know that. This is one of the first outcomes I've seen. You know, this is now 60% drops in the LDL receptor, LDL cholesterol. 60%. Nobody ever gets 60%. If you can take a statin, you're lucky to get 20 or 30%. 60% is huge. Okay? Now, what does it do? Well, it decreases your ability, your tendency to get a heart attack by 50%. That's huge. Okay? So I think this is really going to be a success story. It's rare to get a gene that has such a great effect, you know, but it's now being hailed as one of the biggest discoveries in cardiovascular diseases in modern times. But you can see that we started by discovering a gene. We didn't know what it was doing in 2003. Slowly, we began to see what it's circulating, what does it do, what is the receptor, until finally now, it's on the market. About 13 years later, that you can get an antibody against this protein that's really on the market. The interest in the scientific society is huge, because you can see that in 12 years, there's about more than 1,300 manuscripts just on this subject. And it's growing very fast. So I say to students or to people in my lab, you know, we started with this Pandora's box, you know. We were alone, we were fooling around with our enzyme and nobody cared, you know. And look at it now. There's so many things happening with this enzyme. I don't have time to show you. It plays on sepsis, it plays on viruses, it plays on other fibrosis and so on, you know. But this is what I'm really trying to say to the students in general. Even though this is really considered to be a terrific example of people call it, I love New York, I love PCSK9, you know. But I think this is the most important thing for you as young people and students. When you're working on a subject, never give up. If you really believe that your stuff is good. Here's this guy who was looking for diamonds. He's really working very hard to look for his diamond. He's almost there. I didn't find any diamonds, I'm dropping it. Don't do that. Okay? Don't let yourself be taken away. Just trust yourself and trust your feeling that if you're subject, you believe in it, move on. We moved on it, we were alone in the field, and nobody is down. You know? I think this is it. I hope I conveyed to you what enzymes do, very valid, and how we can go from an enzyme to the real world of the clinic, of the patients. So if you ask me what will come to the exam, I just hope that you will understand what are the possibilities that you can have.